So today what I was going to talk to you about was that moment right when you're about to submit, or maybe when you're looking at proofs and you realize you need to write down the methods for your microscopy experiments. And some of these, unfortunately or fortunately, may have happened quite a while ago. Some of them will be quite recent and undergone a lot of change. So we're addressing that issue, that help. How can you get help from the imaging scientists around you? And what are we doing as a community to help support this? Let's see if I have my mouse. There we go. So I'm just going to go through the workflow that I'm assuming many of you in the audience have had before, where you're sitting at a microscope and you say, finally, I found that cell, right? I better take several minutes to get my beautiful picture that will go on the cover of all of the best journals, right? And then you go, okay, well, I better take another one, right? And which one do I take at this point? Well, when we do that, there's a huge cost, right, in our time and personnel and that expertise spent doing all of that, all of those repetitions. And in general, we actually get very little payout. Of all of the data that your eyes are seeing, very little is preserved and communicated to others, right? So how, as a field, have we addressed this? And I would say many ways. So while that is true, that used to be the way that people are imaging, now many things are happening to make imaging faster and more productive. So one of those, and you may have that there as well, is that instead of standard confocal scanning, you can use different scanning mirrors to do what's called resonant scanning. And that moves the laser at a faster rate. And so you can acquire images of similar quality much faster. What we find then is that if instead of wanting to go faster, you want more detail, right? There are now many ways, either microscopes or post-processing, where you can apply tools and get more spatial detail. And often this means that your images are that much bigger than they were before, or you're duplicating images along your process. And so there are times, of course, when this all combines, and this isn't even a light sheet image, but you can start tiling, doing multiple colors, doing huge volumes with cleared tissues, and even in intact cells. We were talking about lattice light sheet and just the terabytes of data that you start to get from all of this. So with all of this, you get to a point where you start asking questions about bigger data, right? What if you could image the whole slide? What if you could image the whole organism and then select cells very quickly and do the analysis right then? And then what happens, right? If we could actually have a computer do this more for us, maybe I'm not needed for all of that, right? And as we've gone down this road in imaging in the past maybe 10 years or so, what ends up happening is you get microscopes that start doing work for you, right? And our data sets are getting even bigger and bigger. So you get this data, you've gone through more experiments than maybe I even dreamed about doing in my training, and you're starting to write your paper, right? So at this point, you say, what if now we're saving all of it? You're saving your map, you're saving your context, and you're getting out that data almost immediately. And so when we do that, you can, with many different tools, including ones using standard microscopes, using special microscopes, using computational tools that include acquisition and analysis. But all of that incorporates moving your data. So in this workflow, you're dealing with changing data types and movements of data. And so that includes moving data often from your acquisition computer to some sort of a storage where it can be read by the user. From there, we analyze it, we present it, we share it, we publish it, and we need to be able to move this data in ways that make sense for all of these different purposes. And with that comes the idea of data provenance. And so because each step in data transfer or analysis introduces the possibility of it being altered, either because you mean to, because you're analyzing it, or from unintended ways where it's getting clipped or down rendered, it's critical to maintain the original data file, because theoretically from that point, all of the rest can be recreated. But also all of those steps are important to map, and many of them are parallel pathways that your data has taken before you get to publication or before you get to the sharing stage of your data. And so that record of each of the steps that data takes is called data provenance. And with this comes data duplication. And so with that, we generally say you'll duplicate your data at least 4 to 6 times before it becomes that figure in your paper. And so this gets to data storage issues, but also just what is your data and how can we better incorporate and be true to the integrity of that data and its process. So when we talk about imaging data, we're talking both about the image itself, which can be multidimensional in either space and location and color or time. We have metadata, right, that can be read in this case from Fiji, that's telling you about details of that acquisition. And then you can also have biological or experimental metadata that is very critical. What was that cell? What time was it taken? Was it fixed? If so, how? What were the antibodies? That normally are not recorded automatically and then would be in your lab notebook in some way. Your data is often saved in proprietary formats and has to move to that in order to be analyzed or presented. And so because of the way that journals require data to be changed for figures, again, all of this leads to different duplication and possibly data loss. And that's because data can be altered by both conversion and compression. And those are processes that we're working on, talking with our users about, ways that can make their lives easier, but that they understand what's happening to the data as they move it. Now, when we talk about metadata, what happens is each of these companies and each of these proprietary formats have different ways of making their data and their metadata useful for their software. So different ones have different ways of saving that. And so for some, that is so many pages that you could never possibly as a human being read it from a given experiment. In the case of the ARI scan, in some of the others, there's so few lines that you barely know what happened. And then there's everything in between, but it can vary quite dramatically. So although you're told that metadata is often saved automatically, what that entails can vary quite a bit. So often what happens is we don't actually get to this point, right? So one possibility is that you do publish and everything went very smoothly. You followed your data, you published, and it all went very smooth. The other possibility is you get to the microscope and you go, that's not the cell I thought I was going to see, right? Or that's not what I saw those other people do, right? And so we get many people asking us as imaging scientists, how do I do what these people did? And in that case, when we look at published methods reporting, this is why we focused on it. It comes into our lives as imaging scientists in core facilities almost every day, but also to many researchers. And you'll get things like this, that the microscopy, regular, was performed on a Zeiss axioscope upright and images were taken, right? Another one will say that all of the signals were DAPI and that they were acquired with a confocal, right? All of them in the entire paper, right? And this is about as much methods as we can get. So when somebody brings it to us, it's difficult for us to say how to reproduce that experiment. And if you go to the figures, that's normally our second stop, right? We go and we go, okay, well, the methods might not have been complete, but let's look at the figures. And what people have found, and this is a figure by Helena Jambor, is that the figures themselves are also often not including all of the information that you would need to recreate that experiment. So first of all, they may not be communicating data very efficiently if they're not colorblind friendly, but also scale bars may be included without the reference scale. Insets or other things are not annotated at the way that you would expect to be able to know where this image came from and how you could recreate it. So a paper came out that really had a large effect on our field by a group at University of Minnesota. And what they did is they looked at biological journals and they went through all of the papers and decided which ones had any figures that included a microscopy image. And you can see that the fraction of articles including at least one image from a microscope was quite high, from almost all of them to very close to 50% or more. Then what they said is they took a protocol of deciding whether or not it could be even feasible to recreate those experiments by looking at the methods sections. And what they found is all of them failed that test to a great majority. And at most, one of the journals got about 50% of their articles could have had enough data to reproduce those experiments at a broad level. And so what does that mean? Why are we concerned with how methods reporting may be incomplete? Well, partly it's because people don't know why that could be a problem, right? Why would not representing what objective you used or what camera change the interpretation? And so part of this is education, and we're trying to work on the why, the how this technology can be best used and interpreted. We also have the space limit, which has been increasingly difficult, even though we're moving away from print journals, where one of the first places people cut is often the methods, and then it's put into supplemental material where it's not reviewed often by reviewers and editors and not formatted or checked to the same rigorous levels. And then often there's just a lack of awareness from others who have tried to reproduce experiments like this or technology, and they don't know the amount of detail that's required and why that affects the interpretation of the results. So I want to take you through a recent paper in collaboration that we did, and it was predominantly done by 4 different groups, led by Paula Montero Lopez, who's at Harvard Medical School, and then our group at UNC, Lisa Cameron's group at Duke University, and Mark Sanders' group at the University of Minnesota. And that was the same group that published on determining the methods reporting in those biological journals. And another figure showed that only, I think it was 50 words on average were dedicated to describing methods for microscopy. They actually quantified the number of words that were used, and they were so low. So I just want to talk to you about this because the focus here is for the group like you all are on educating our community and everybody doing microscopy and making it easier for you all to both interpret your own imaging data and others and to report it for others to use down the line. So first is kind of the why, and that's the main meat of this paper. So for example, how does your illumination selection affect your results? So this is showing a bacteria that's being imaged. It's expressing a GFP, but it could be imaged using a green wide field filter or a yellow, right? And many of you have found this where you can image the same fluorophore and you can put it through a multi-band pass filter, and then you can fit more colors in. Right. And so some of our microscopes are set up for that, but that means they're not optimal for a single color. In this case, what you see is the green is brighter, and that's the graph on the right, than when it's acquired with the yellow, even though we see signal. But what does this do to the data? This means that if you look closely, the data in green is more robust. We're able to see structures and our contrast is better. Whereas with the yellow, even though it's the same signal and we are collecting it, our data quality is lowered. And so we actually don't see structures that are the same and could be visualized if we were collecting more of that light in a different way. Now, this is on the other side, right? What if you don't know how the light was collected? So in this case, on a confocal microscope with DAPI, you are illuminating with the same laser, the 405 laser, but you can pick the window that you're detecting it in to be wider or skinnier. And so what you're seeing on top is in the gray is the excitation profile for DAPI, and then right shifted is all of the light that DAPI is emitting when it's excited. And then in this cell, it's also expressing 488 phalloidin, right? And so those are the green is the excitation profile for 488, and the greenish yellow farthest right is how it's emitting there. What you can see is when you collect light in the 415 to 475 range, you're mostly only getting DAPI, even when you turn the laser up. But if you widen that, again, like you're taking more of a long pass filter, even though you shouldn't be activating 488, you start to see structures that look like the phalloidin in your DAPI image, right? And we often get this question, how do I know that I'm not getting crosstalk or bleed through? And so there are different controls you can do, but this is the risk of not reporting what those laser intensities and laser lines were used to collect, and then what emission was collected very specifically, because you can get very different results and even see structures that are there, even if they're unintended and not the target molecule. So similarly, and what you've probably heard of if you've taken any imaging courses or you've done some training on the microscopes, is the objective lens can be very critical to how your images are created and the data you get out. In this case, what you're seeing is a 40x lens that either has a 0.6 numerical aperture or a 0.95 in the yellow or a 1.3 in the purple. And what you can see is, first of all, if you do the same collection of the light, emitted light, the lower numerical aperture lens is less able to collect that light, right? So it appears dimmer. So even if you compensate for that and you look at then an inset of that image, you can see the image quality is degraded when you have a lower numerical aperture lens. And so you don't get as much contrast, you can't see as many fine details in your structures. And that can lead you to make different biological assumptions of whether or not something is connected or one or two that you don't have because you don't have that spatial resolution. So if you don't know that and you're making your analysis or in your interpretation based on that, it can be very both difficult to reproduce and difficult to interpret at what level you're able to make your biological conclusions. So the other thing I wanted to bring up, and I'm not going to go through all of the data in it, is that some of the things that our users don't often think about is that experiment order can affect your results and the integrity of your results. And what we mean by that is, in this case, are you taking your Z stack with multiple colors in one color Z first and then another? Or are you at each plane taking two colors and then the next plane taking two colors and the next, right? So whether or not you're doing Z depth and then color, or you're doing color and then Z, can affect your precision. So stages are not always able to return to the exact same depth with as much accuracy as when we change the color turrets or change the emission and detection windows on a confocal. And so what you're seeing here is when you take a Z-stack first in one color and then in another and overlay that single point, you can see that the red is above where the green is, right? So there's an offset. It hasn't quite overlaid as cleanly as when you take red and green in the same color at the same position and then you change the Z. So depending on what you're wanting to measure, that can affect your precision in a different way. And we find that imaging order of experiments and collection is very rarely reported. And so that can often be something that the user is also troubleshooting when they're starting. And so if you think about when somebody else in your lab is going to use the microscope for something very similar, many of the things you tell them to do are not reported in your methods of your final paper or in a full protocol, but can have a very deep effect on whether or not you're able to use that data to answer that biological question that brought you to the microscope. So we made a poster so that this could be easy to reference. And for us, something where we can address it to different stages of the imaging and different specific hardware to let people know what does it look like when you have different numerical aperture lenses, or what does it look like when you have different corrections for chromatic aberration, or if you don't. And so when they see something on their microscope, they can look and see, is that looking like this? Could that be due to that? And if so, how might I control for that or adjust for it? And so all of these images are also in the figures of the paper and go through many more details just to show people how things can be affected by these reported details. So if you don't know them, you might actually get a different result than if you did a different setting. And so we hope that this helps you all to understand your hardware better and more fully, and that as you're setting up your experiment, you think about some of those controls and where you may be getting either the ability to more focus on the structure you're interested in, or time point or precision with more clarity, because you understand how that light is going through each of the different decisions you're making during acquisition. And this paper itself focused on wide field imaging, single point scanning, confocal, multiphoton, and spinning disk microscopy. So we didn't get into all of the different modalities, but we hope to keep adding to it and hope that others will too, as we get more specific applications as well. So the output of that paper was also several tools where we have PDFs of checklists. So when you're writing your methods, you can go and say, I did a wide field experiment. I should report what the stand was that I used of the microscope. So that would be like a Nikon TI2, for example, or any of your favorite brands. And then it asks other things. If you did multiple positions or live, there are other elements of the stage that it will prompt you to suggest. And things that are in italics are recommended if you know them, but not required. And that might lead you to also ask your imaging scientists or your vendors about what this means. How could that affect your data? What is the one that's actually on your microscope? And then in that PDF too is also an example of an example text that includes all of this. So it doesn't need to be done linearly based on the checklist. And so we included several different examples in this of different ways people could include all of this information that was required, but not be pages and pages long, right? And with this, it's all linked. So if you click on one of them, you will get examples to it and let you know kind of what we mean by that. And then that will bring you back to the checklist if you want to return to it. So it's all jumpable, if that makes sense. So that's great, right? But we did this with some of our students and they were said, well, it's still really long, right? And I don't know what the focusing device range is. So how do I know if I need that? And so that prompted us to add another tool, which is called MicCheck. And this was really led by Rebecca Senf and Paula Montero Yopes at Harvard with this exact question in mind. So what she did, and this was coded by Rebecca, is it was driven by the idea that we wanted something really user-friendly and accessible to everyone. So it would be web-based, it would be based on simple questions that anybody who was acquiring that experiment would know. And we're going to focus first on fluorescence microscopy, knowing that there are many other modalities that this could then be applied to. And later, we also wanted this to be very flexible and adaptable in ways that those PDFs obviously aren't, right? So instead of being pages that you're crossing out, what can be done for my specific experiment that I did today? And so for this, the first version of MicCheck was in that paper, and it dynamically generates a checklist online for your methods experiments. And so it asks you simple questions, and based on that, it will include or exclude elements on the checklist that should be relevant for your experiment. And then you can toggle a thing to see what examples of that are. If you say, oh, I need to know the objective numerical aperture. What does that mean? It'll show you in an example format. And then those checklists that you just built are downloadable for you as a PDF, so you can save it, print out multiple copies, and put it in your lab notebook if you wanted. So what does this look like? If you go to that web left first, and those are those questions. And as you change and answer those, so you tell it that it was a wide field experiment, you'll see elements appear and disappear from the checklist on the right. And as you go down through your questions, it will build that checklist specific to whether or not you did multiple colors or a time lapse or tiling or a live experiment, and not include the things that weren't relevant for that experiment that you're reporting on right now. Oops, now it's highlighting it. And so the use cases for this, who are we planning that this might be useful for? It's first any scientist that uses a microscope. And that's for both planning your experiments, so that if you did this before, you could think, oh, wait, maybe I should talk to people about what speed I should move the stage at. Because if I move too fast, maybe my sample will come unmounted. Or maybe I should think about if it's more important for me to keep the colors in Z consistent in time and collect that fast, or if I should do multiple colors over multiple Zs. It can be useful for writing grants because you know what equipment you need to be able to do that level of experiment, and for writing up papers because you can check the methods that you've then written to make sure that you've covered each of those components. We hope it's also useful for core facilities and for journal editors and reviewers, because it can be a resource that you can talk about with your users to help them understand, as you're pointing at and changing things on the actual scope, they can see why gain is important, or laser power, as you watch bleaching maybe happen or not. And then for journal editors and reviewers, it lets you run through when you're editing a paper right at the end and you've commented on the overall experiments. You can say, wait, in order for this to be reproducible, you're going to need to include what order you did this in, what objective you used, and what lasers were actually used or filters for this. So it can make that in a much easier way where you don't have to sit through and think of each of those details on your own. And so, speaking of core facilities, I wanted to bring you to the next phase of this project, which is MicCheck version 2. And so this is something that Rebecca is actively working on and that I'm personally very excited about, is to build this for specific microscopes, where you select it was the 20th objective, and I know what that numerical aperture was and who the manufacturer is. So all that text be imported automatically for all else. It will be accurate way. And so that way people can use cheat familiar with. Often microscopes have specific name. Workflows can have or microscope specific prompts that somebody knows. So the microscope that was and which objective was, because it has a nickname, and that can be put onto these checklists as well. So this would allow cores or labs to upload their own unique metadata, so you don't have to look that up each time, and it allows you to store it in a way that currently the online version of Check doesn't you do. And it allows you to customize those to any extent that you would like, to have a separate output, but you could add in biological data if you wanted, or things like that, to be more and more useful for the experiments that you're using. This is ongoing, and so all of this is up in GitHub, and we really value feedback and contributions, and I encourage you to reach out to Rebecca or Paula if you're interested in this. We're moving to the stage of trying these out in our facilities with MicCheck 2, and it'll be really interesting to see what people find useful and what they find to be bottlenecks. So any of that feedback is always welcome. And so this brings us to what comes next, right? And it's the writing. It's the generation of that text. And how do you get from there to different styles of reporting that accurately? And then as we were doing this, and many of you have done this probably, the thought comes, couldn't a computer do this pretty well, right? I mean, I still want to check it. I want to tell it what to do, but maybe this could be more automated. And this leads us to another project that was recently published called Methods J2. And this is following up on Methods J1, which was by Mark Sanders' group. And it's a software tool to capture metadata into a draft methods text that is written in sentence format that then you can just edit for your needs. And so in this case, what we have at the top and what we just talked to you about was a checklist, which we hope will affect how you're acquiring the data. And also another tool, which I just wanted to introduce very briefly, which is called Micrometa, and that's led by Caterina Strongio de Castilla. And that is a way of keeping a computer file, a JSON file, that includes all of the details of all of the hardware with its transmittance levels, all of its manufacturing, all of the threading, all of those things that are specific to that piece of hardware, but most people would have to look up and know. The methods checklist can help inform what is required for that. And then what you get from your imaging and the metadata that you save, right, after you've taken your experiment, is then methods J2, that script, which will then write out methods text for you. And that will also be guided by both the user and any imaging scientists you have that help you to fill in that hardware data and make sure that you're reporting that proper way. So this is what methods J2 looks like. It's a image based macro. And so when you run it, it asks you to select an image that you want to write the methods on. And so from that, it pulls out the metadata that it sees. So you don't want that to be your final processed image. You probably want that to be your raw image. And it'll show you the metadata that's opening as well. And then from there, it'll take you through and ask you questions about your acquisition. So it'll ask you things like how would you describe the system that you used? In this case, they saw from the metadata that it was a Zeiss laser scanning confocal, but it just wants to make sure from the drop-down menu that you would consider this a confocal laser scanning microscope. And then there are other checkboxes to include other experimental details, like whether environment, select things like your objective. Again, it will say if the metadata is there, what it thinks it is. So it's right there for you to reference, but then the entire name that you would report in the text is then there for you to reference in the dropdown. And you continue to go through this to recheck those steps and make sure that that metadata is accurate for the acquisition that you actually took, and that hasn't been corrupted or lost in some way or need to be edited. Then at the end, what it'll do is actually give you some example text, and that's what's shown here on the bottom, where it's in full sentences and will tell you an example of how you could report all of these equipment data for your methods of acquiring that image that you opened. And then of course that can be looked at by the user, make sure that it makes sense, added to or changed in a way that feels best for that specific experiment. So there have been many other articles that have been written about different stages in microscopy and how to report it, and then how to make reproducible data, how to share reproducible data, etc. And I encourage you to look at all of these and to continue to contribute to the field, to let us know what is working, what is not, what you wish was there, so that we can all work on this together to make it easier for everyone. I just wanted to thank everybody there and all of you for listening, and all of our collaborators. I tried to include the authors as we went, but wanted to list them here as well. And then those highlighted either contributed slides or were first authors on some of the papers I mentioned. And then I just wanted to acknowledge my core team as well, and those grants that help support our core and these projects that I mentioned here today. So I know that was kind of a whirlwind, but hopefully showed you a few new things that we've been working on. Thank you all so much for your time and for being here.
Thank you very much, Michelle. Great seminar. Fantastic introduction to all the tools and all the different things that we have to take care in order to be more reproducible in our experiment. I really enjoyed the seminar, and we have a lot of information. But just after we finish the seminar, I will start to install the MicCheck in all the microscope we have around, and
it's just a website. You can
start to play around with the checklist. It's very nice.
And it's just an Excel sheet that you can edit to upload to make those checklists custom to your own core. So we're hoping that saves people a lot of time and accuracy. I can stop sharing if we want to see people for discussion. Would that be good?
Absolutely. So if there is anyone that would like to make a question, I also want to say hi to Lisa, and thank you very much for joining us today. You can raise a hand, and then we will basically open your mic, and you can be able to do your question by your own. Meanwhile, the people, thinking question, I really think that one of the very early things you mentioned today about, these little things about knowing the instrumentation you are working and understanding, how are the best regimens that the instrumentation are working is so crucial to eventually take advantage. You push a little bit more of what usually an instrument can give you. And this simple example, of using, the green versus the yellow, how much change and how you can detect different things. And this is basically something that if you understand, that the hardware has different, profile of sensitivities and, the brightness of your protein, this protein has more brightness than the other. And if you, understand this detail, even before to start, to cloning your molecule of interest with the protein of fluorescent protein and everything, you can build up your experiment with more, with a lot of more, support and a lot of more, opportunities for whatever you are going to look. And I think this is really simple example, right? If you move in the range of your detector, okay, your detectors have a profile, it's not flat. If you select a red, it will be less sensitive to green. Signal to noise also will be, another issue. So I think this is very important for people to understand, right?
I know I was shocked when I realized, and I should have, but I went through my whole training and I didn't realize that the mercury lamps I was using had very specific peaks of excitation, and they were not all equal in intensity. And I should have known that, right? If I had been thinking of the chemistry or otherwise, but that's why often my red was so dim. It had nothing, I was blaming it on my fluorescent tag or expression, but often it was because I was hitting it with much less light to begin with. And I hadn't even thought of that, right? And so I feel like there's so many of those that we take for granted and we assume are equal. And that it's only when we see that when we go, oh, we're so good at compensating, right, in other ways and adjusting our experiment that you don't realize what you might be missing or assuming, because they're kind of silent assumptions we do. And the other thing that I've found is that the protocols can take a long time to write, but when people do, they often lead to publications. You can either put them on free free and open source things like bioRxiv or protocols.io, but then there's also many published ones where you can go shortly from there to publication. And the amount of that saving people time in many labs has been incredible, where now once somebody put that time in to do it, it's so much easier for the next person who comes in to understand what needs to be done, because there's a video of them actually mounting it, or a screenshot of the acquisition window that includes so many more details than you normally tell a person when you describe the experiment to them.
Yeah, another important or interesting point of discussion that you also bring during the presentation is, this compromise that journal has nowadays with the word limit, right? How many words you can use or not. And you are editor in chief of biotechniques, so you are every day handling, with these difficulties that you need to have, a limit of word. Otherwise, the paper will be, over and over, big. What do you think we can do? Because what you say is absolutely true. I mean, if you squeeze of the material method in the main document and you move to the supplementary material, most of the time it's not well reviewed. Most of the time the people is not even aware that it's there, and basically you are losing. And this is something that, when I have chance to review papers and they cite other papers, I always complain, because I really like to see in the same document what is going on. I don't want to read 2 or 3 other papers in the back to understand what they were doing or not. So what clever idea we can come up, with the journals in order to, have this information there, right? Be open in order to belong with the methods.
That's my hope, is that some of these things will become linked, right? Where for the average person who wants to look at the findings of the paper, assuming it's gone through peer review, you may not need all of those details, but anybody who is in the field will need access to it, right? So I think what's important is that we do it when people are still fresh, right? So that it's linked to it, because right now you have the ability to link a protocols.io or a star protocols to a paper, but often that can be done years after the fact. And then as you're reviewing a protocol, if you ask for something, they're like, well, that's already been published. So yes, that's a better control, but I'm not going to do it that way, right? So I think there's an issue of doing it after the fact too, even though it's helpful, it's not as if we can do it with the paper itself, when actually the experimental data can be verified and strengthened by it. And so I think what needs to happen is, it needs to be easier for the reviewers, where I know I did that for one of the ones I was doing, is I printed out the checklist so that I could check myself. Oh, wait, I should ask about the stage. I didn't think about that, right? Because I was so intent on how they cloned their reporter or something like that, right? And so it helped me go through something quickly, not having to do the work to inventory all the information I might need. And that also means that others who aren't experts in that might be able to at least know and ask the question while it's still in review. Wait, I noticed you didn't say what order you collected this in. Can you, please elaborate on that? And I find that once you do that in review, people really do take time to do that very thoroughly. It's just whether or not they were prompted to do so. So I think it's making that easier and more relevant, right? Not making it tedious for people. And then having it be relevant to the point where we're really only asking if there's another option for that would make that data not interpretable, right? If we don't know any objective that you used, I can't tell if that was a tiled image or if it was done on a stereoscope at 0.6 zoom. I need to at least know some of those details to know if you can measure structures that size.
Yeah, right. Yeah, this is another role of the core facilities, right? We are around to support in these little details that eventually, users eventually miss or don't understand at deeps how important can be, for the reproduction of experiments. So yeah, I absolutely agree. So I don't know if I question, Andres.
Hi, Michelle. Thank you very much for a wonderful seminar. Very important for all of us, for all the community that uses microscopy to rediscover these concepts that I think are always a problem for reporting results and for actually planning experiments. For the whole young community, it's very important to realize that there is a way more in the microscopy images than just going and take a picture, and this can be very impacting the results and the science we do behind it. So this is thank you very much. I just have a question, like very kind of silly question in terms of functional things in the unit. Like you have a role in teaching the people to learn these things, right? But then they come and say, okay, we did these experiments. How do you follow when they say, okay, we did these experiments in the unit of, in your unit, for instance, in your core facility? Do you review what they publish, or I mean, when they mention you? Because it's somehow they are saying, okay, we have this kind of standard of quality because we did it in your units, but I guess you cannot be following all the papers that come out of there, and so you don't know if the people is actually saying what they should say. How do you handle that?
That is a great question, because right now our university has no say really in what publications go out. There's no way of reporting to anyone who is an author on a publication, who was acknowledged, what grant sources they used. That's blind unless you report it, right? Or the journal does. So right now, the way that we do reporting to help measure the impact of the core is linking to grant numbers, but we also made it's called an RRID number. It's a unique identifier for our core. That means if people reference that in their acknowledgements, it is linked in a database to any of those. And so that has helped us track some. Otherwise, it had been, looking at our main users manually at their publications and seeing if we contributed. And then it's almost a coin flip. If we go in, were we acknowledged? Were we not? Were we acknowledged properly? And then it's kind of a communication relationship building on how do you address that, and how do you, if you were acknowledged improperly or methods weren't properly reported, how can you help educate them in a way that isn't too burdensome for them, right? But that can help to create the scientific record integrity, but also, say, well, actually, you said you used a microscope that we don't have in our facility, right? Or a camera on our confocal that doesn't exist, right? And there's no check for that. Sometimes people send them to me, and then I'm able to help add in information, and almost always I get, that was great, I'm gonna save that for mine. It didn't quite make it all in the paper, right? But at least then they've had some of that already given. And then I think the way that most people train is they look at previous papers and they look at what was reported and they start to recreate that. So I think as we start to address it and it becomes more of a habit and it becomes kind of in the template, that people will have an easier time editing it. And so it's getting it to that level and then getting reviewers to prompt for it, or grant reviewers, paper reviewers, editors to say, hey, you need at least this reported. And then as people learn and see them in the papers, they'll just naturally start to reproduce them. But yeah, right now there's a lot of error in the scientific record there because there's no validation. Even if they acknowledge us properly, it doesn't come to me to look at the methods or otherwise. So we have talked about that as well. And some are bringing in what they call scientific consultants as reviewers to just look at specific portions, which can be hard to do without the biological relevance, but you can envision they've done more for sequencing, and they send them that and like, does this make sense? Can you at least check quickly that these primers are even possible, right? Or is there a huge typo in it and that should be corrected? You could do something like that for microscopy. It's just, I feel like the review will end up including a lot of questions. Did you use this? Did you do it this way or this way, right? But so it won't be quite as cut and clear, but I think there is some talk of doing that. So it's not as burdensome on the experts, but also increases the integrity before the publication happens.
Thank you.
Thank you, Michelle. Any other question from here? No. The
From
chat,
the audience?
there is no comments. I don't know, before to close, if Lisa want to do any comment?
Thanks for having me. I think most of what, all of what Michelle says, obviously, is I was part of the project, and we have the same kind of problems too. And I don't claim to have any better, I guess, compliance of people following the methods. I have, usually it's, I don't know, 1 in every 10 or something like that. And it's hard, keeping track of publications is still difficult. We mostly know when people tell us, or I start, there's one link in here at Duke, because there are many many labs that use our facility that are part of what's called the Duke Cancer Institute, and they have a mechanism for keeping track of their publications that have to do with the Duke Cancer grant that they have. So, but the same kind of thing happened where Michelle just mentioned, where people will say, oh, I used the microscopy core, and then I go and look up the paper and it describes a microscope that we don't have. I'm like, I don't think you used our core for this, or you wrote it down wrong. So I think it's just continuing education to try to get people basically to talk to us earlier. I had someone say, oh, I'm writing this methods, and I helped her write it, and she's like, oh yeah, I'll try to get that in when the reviews come back, right? So
Yeah. So excellent. Well, thank you, Lisa, for also joining in the seminar. Well, I would like to really thank Michelle for this kickoff. It was a wonderful seminar. I think all the people enjoy a lot. We collect a lot of information that we can read and we can go through. And for sure, this is the first of another 3 or 4 seminars